Our body is made up of billions of cells. These cells use various proteins to help them perform vital functions, from processing nutrients and eliminating toxins, to regulating physiological reactions and defending the body against infection. Many diseases are caused by malfunctioning or missing proteins in the body. Advances in biotechnology have led to the ability to use living cells to produce proteins to treat some of these diseases. Genzyme uses long-established, proven techniques to modify mammalian cells by equipping them with a piece of human DNA so they can produce therapeutic proteins. The proteins produced by the cells can be used as targeted treatments or to replace malfunctioning or missing proteins in the body, addressing serious, often life-threatening diseases. The modified cells are grown and carefully frozen in liquid nitrogen for future use. This collection of cells is called the cell bank. Compared to the highly automated manufacturing of chemical drugs, which takes just a few days, the production of therapeutic proteins is a complex and closely monitored process that lasts several weeks or even months. This process starts with the thawing of a vial of cells from the cell bank. The cells are mixed into a carefully prepared nutrient solution called the growth medium, which feeds the cells and lets them grow and reproduce. Each cell produces a tiny amount of the specific therapeutic protein. Over a period of weeks, the cells will multiply from several million to more than 30 billion. This cell population of billions of cells is transferred to a vessel called a bioreactor, which provides sufficient space to allow the cells to continue multiplying. Once they reach the full capacity of that bioreactor, they move to a larger one to continue the growth process. This is repeated several times until, weeks later, there are enough cells to go to the final production bioreactor, large enough to hold thousands of liters. Once in the main production bioreactor, the cells are fed additional medium on a regular basis to allow them to continue growing and producing the therapeutic protein. The content of the bioreactor is regularly harvested and processed to retrieve the proteins produced by the cells. The cells meanwhile remain in the bioreactor, producing more proteins. Every day, samples of the solution in the bioreactor are taken and tested to monitor the growth and quality of the cells. The harvest solution contains the therapeutic protein along with other proteins and substances. It is taken through a succession of special filters to eliminate unwanted compounds and obtain a batch of purified proteins. These undergo strict quality control to provide safe, effective treatments for patients. A treatment course for a single patient requires several milligrams of purified proteins. To produce only one dose of medicine, several thousand billion cells are required. The purified protein solution is filled into small vials in a sterile environment to prevent contamination and ensure the safety of the product. Some therapies go through a freeze-dry process. At the finishing stage, each vial is inspected individually for irregularities. Samples are taken through a final quality control to ensure product purity, safety, and potency. The vials are labeled and packed before they're sent to treat patients around the world. The production of medicines faces demanding safety and efficacy standards. Over the years, Genzyme has worked continually on the development of innovative technologies to produce reliable and effective therapies. Genzyme focuses on medical and technological areas where it can make a major positive impact on the lives of patients with serious diseases. Genzyme's therapeutic proteins provide treatment for thousands of patients around the world.